Okay, so let's talk about mutations. In general, there are three main kinds of mutations. There are base substitutions, insertions, silent mutations, and deletions. So whenever I say base, I mean as in one nucleotide of your DNA strand. Your DNA strand is made up of a lot of different bases, and bases are the nucleotides that make up your DNA. Base substitutions are mutations that only change one base of your DNA, and it's also a broader category for point mutations, missense mutations, and nonsense mutations. Point mutations are where only one base is affected, only one nucleotide is affected, such as a substitution for another base. So, for example, if adenine went with thymine, the A and the T are supposed to pair up together, but with this kinds of mutation, the A might be substituted for cytosine or guanine, and it'll pair up in like the wrong spot. The sequences of your DNA, every 3 nucleotides, they code for proteins. And those proteins are basically what helps your body function. They're the ones who carries out the functions. The DNA is just like a transcript of, what they're supposed. They're a transcript of. The sequences of your DNA code for proteins, and so those proteins are basically what carries out your body functions, and your DNA is just a transcript on how they're being produced. A missense mutation is when a point mutation results in a different protein being coded, which it could potentially lay harm to your body functions. So like missense mutations, nonsense mutations alter the protein produced. Although nonsense mutations stop the entire protein production. They stop the entire chain because there are only so many real protein combinations, and some, they just don't work. Moving on to silent mutations, they're the ones that aren't noticed because they don't affect you. So they're also referred to as neutral mutations because you don't notice them. They don't affect you in a good way, they don't affect you in a bad way, they're just there. Insertions and deletions can be either point mutations or frameshift mutations. So frameshift mutations, what they do is they shift the entire protein chain forwards or backwards one nucleotide. And because the entire thing is shifted, they'll dramatically alter the protein production. So a lot of things can cause base shifts. So for example, during DNA replication, the DNA polymerase, which is something that essentially builds the new strand of DNA. So during that process, it can sometimes get confused and it can attach the wrong base. So for example, if A used to go with T, now it might go with C or G. Like we said in our last episode over genetics, mutations can be caused by a wide variety of different factors. For example, environmental factors. So cancer is an example of a mutation, but cancer is caused by, let's just say skin cancer, skin melanoma. It's caused by the sunlight. So the rays can make the cells mutate in your skin, so that's where you get things like skin cancer from. Radiation plays a huge part in genetic mutation diseases. So for example, in Japan, after they dropped the atomic bomb, the radiation around there caused horrible genetic damage to a lot of the people who were around that at the time. So mutations can either be good for you, they can be bad, they can be neutral, like nonsense mutations. So a way that mutations could be bad is that they could cause a lot of not so great diseases like sickle cell anemia or Huntington's disease. So sickle cell anemia is caused from a defect in hemoglobin protein, which is essential for your red blood cells. And when there's a defect in this protein, it'll weaken the red blood cells and it'll cause them to take on like a crescent shape, almost like a sickle shape from its name. These weakened cells, they hold less oxygen. Some can't even hold much oxygen at all. They clot easily, and they also have a shorter lifespan. Huntington's disease is caused by the synthesis of too many Huntington proteins, and so because there's so much activity and so Huntington's disease, it's caused by the synthesis of too many Huntington proteins, which essentially is saying like there's too much activity of those proteins. And Huntington proteins, what they do is they function inside your brain. And so when there's too many of them, it'll affect the brain nerves, and eventually this is going to kill off the brain cells. This is going to damage your brain, and it can lead to dementia and brain degeneration. Dementia usually happens in older people because as with age, your brain gradually wears itself out. But Huntington's disease can cause early onset dementia, which is not. So eventually this will kill off the brain cells and can lead to dementia and brain degeneration. Normally dementia happens in people older because as with age, their brain gradually just wears itself out. But with cases like these, it'll be like someone in their late 20s or early 30s. So, and this form of brain degeneration can cause, depending on what part of the brain that it's affected most, it can cause things like you could lose some of your motor skills, certain sides or certain parts of your body might become, disabled and difficult to manage.